It's a pleasure to be here, and we thank the organizers for the invitation to be part of this event. Maarten UYTTEBROEK, my colleague, and myself, we come from Belgium. We work at VITO, and VITO is a research institute focusing on applied research in different fields. And one of these fields is also sustainable chemistry, and we are, let's say, working more in the field of fractionation and extracting different types of biomass, being insect biomass, but also side streams, algae, and so on. Today, the subject is the BBI-InDIRECT project, and in the first part of this presentation, I will try to present to you the general thoughts behind this project and the aim. First, some facts. It's Horizon 2020 Research and Innovation Action that was granted within the BBI call on innovative, efficient biorefinery technologies. So the focus is on biorefinery technologies. And as mentioned here in the title, we look at different biorefinery approaches called here direct and indirect biorefineries. It's a project of 3 years, and it's coordinated by VITO, and as coordinator, I am pleased to present you this project in name of all the partners. We have 9 partners from 4 countries: Italy, France, the Netherlands, and Belgium. The 2 research partners are the University of Parma and VITO, and then we have 7 industrial partners, among which there are 5 small and medium enterprises. These are Nutrition Science, Millimeter, Improve, Chemstream, Innovatiesteunpunt, Temperio, and also Protifarm. Our story starts with side streams. The aim is to convert side streams into marketable compounds. And as you can see, a variety of side streams was considered initially. And to extract these compounds, there are different approaches. And the first approach is that for each of these side streams, or groups of side streams, a separate biorefinery is being developed. And this is what we call the direct biorefinery. Now, this can be challenging because we are looking here at side streams that can be relatively small in size, that are geographically distributed, seasonally dependent. So it can be a challenge to construct an economically viable biorefinery that can be operated during the whole year. That's why we also looked at another approach, the indirect way, and this is where the insects come in. We see the insects here as the first step of our biorefinery to convert this heterogeneous potentially mixture, let's say, of side stream into one homogeneous biomass. And this biomass is the insect biomass itself. So the insects convert the nutrients in the side stream into their own biomass. And the next step is then to develop a biorefinery to convert the insect biomass into crude fractions and purify them further, envisioning proteins, lipids, and chitin as the main fractions. Both biorefinery approaches, we see them as complementary because they can produce other fractions. I forgot to mention, but for the indirect biorefinery, of course, we also have the FRAS, which we call here the enlight compost, that can also have some value. So both are complementary. In the end, we end up with several fractions that can then be formulated into products for different application fields. Some are mentioned here: food, feed, chemistry, agro, and the cosmetics. All legal and policy issues are important. We do take them into account in our project in the sense that we make an inventory so that we know where the bottlenecks are, but in our research we do not feel limited by this legal framework, because in research we have to work for the future where legislation can be changed. The focus of the indirect, so in the project we compare the direct and the indirect biorefinery, but the main focus is on the indirect part. And our partners, you see, they are active along the whole value chain. We have a partner dedicated to collect and identify the side streams, and they are supported also by Improve collecting this. Then there are 2 insect farms, Protifarm and Millimeter. VITO is focusing on the biorefinery. Then for characterizing and further converting the fractions, also the University of Parma and ChemStream and Improve are involved. And then towards the end applications, our main focus is on feed and chemistry, where we have nutrition science as a feed specialist and ChemStream for the chemistry. So to summarize the general objectives, we are focusing mainly on the indirect cascading biorefinery approach, consisting of 3 steps. First, the homogenization of the side streams, so converting it into the insect biomass, then fractionation of this biomass in crude extracts, and further purification and conversion into the products. We compare it with the direct biorefinery. We want to optimize this biorefinery in terms of conversion efficiency and also maximal valorization of the feedstocks. We work towards compounds, so different application areas are explored with the extracted compounds, and we take into account the whole value chain, implying economic, environmental, legal and practical aspects. We work at lab scale up to small pilot scale. The challenges that we want to address is management of waste as a resource, accelerating innovation and market uptake of bio-based products. We contribute to looking for alternative sources for proteins and also alternatives for antibiotics. And the reduction of the dependency of the European economy on non-European countries is also a point of attention. More specifically to the indirect biorefinery approach, the objectives are given here. So we work with side streams. They comprise plants, but also some manure. For the conversion, the aim is to convert 30% of the side stream into the insect biomass and have a 90% use of the side stream when you also include the end-light compost, so the frass. Then the insect biomass, the aim is to convert 70% to marketable compounds, products, lipids, proteins, chitin or others. In the expected impact, we work towards a year-round biorefinery, so there needs to be a smart selection of the side streams here. And on the longer term, we aim to have chitin or chitosan oligomers available at the market at commercial scale, and also to contribute to the production of stable production of recycled proteins. As all projects, we are organized, the work is organized in work packages. The first one is dedicated to the side stream selection and the legal aspects. There is direct biorefinery, indirect, and then we move along the value chain. I will quickly walk you through some aspects of this work package. It's a 3-year project. We started about 1 year ago, so we have a little bit more than 2 years ahead. For the side streams that are considered, the selection criteria, they included the availability in Europe in tons per year, seasonality, also the composition and the dry matter content. It needs to be suitable as insect food. And we tried to find as much as possible underspent side streams to come to a lower cost price. Some of the selected streams so far are depicted here. So there's apple pomace, leek, sugar beet leaves, rapeseed meal, DDGS, and some others are also being considered. For the direct biorefinery, this is the part where there are no insects involved. Just briefly to mention here we focus on leek and sugar beet leaves, and we take alfalfa as a reference material. We characterize these, develop a biorefinery focusing on proteins and also some minor compounds. And we look also at the preservation of the side stream before use, so how to preserve it after harvest and before use. The core of the project is the indirect biorefinery, where we, in the first part, look at the conversion of the side streams into the insect biomass. So questions we want to answer is what are good side streams, what is a good feed for the insects, and what is the impact of the feed that the insects receive on their composition. And the second aim is then to develop a biorefinery for the insects to fractionate it into proteins, fats, and the chitin. We really focusing on cascading biorefineries, so multiple compounds from one source. And there is attention for preservation of the functionality during the biorefinery, especially for the proteins. More details will be presented by my colleague in the second part. Insect species considered, it's black soldier fly, cultivated by millipeter, where there is more, that grows on a more wet side stream, while the lesser mealworm, produced by Protifarm, is growing on more or on drier side streams. And later in the project, also the house cricket will be considered as an extrapolation case. We dedicated a separate work package on co-balancing feed, so the insect feed, and also the safety aspect. So for the co-balancing of the feed, as has been mentioned, it's important to know what do the insects exactly mean. How can we meet this by a mixture of side streams? We want to have the conversion efficiency by the insects as high as possible. And if we find that there's a correlation between what this is the composition of the feed and the composition of the insect, this can also be used to tailor the composition of the insects. For the safety aspects, and this is a part that especially the University of Parma is looking at, is a different parameters will be measured, not only on the insect biomass, but also on the side streams. For the application and formulation tests, towards the feed, we aim for 3 new ingredients that can reduce at the longer term the antibiotics and also imported proteins in the feed. And for the chemistry, and this is mainly based on the chitin fraction, we aim at 10 new building blocks formulating or leading to 20 new formulations. And in this work package, there will be also some application tests as small scale before another work package. The most promising value chain will be upscaled. This is a value chain based on black soldier fly and lesser mealworm. And this part also contains a techno-economical analysis. Then I end again with my figure, which actually summarizes what we want to do in the project. We are now 1 year in the project, and up to now we focused on selecting the side streams. We are working on the biorefinery development in the direct as well as in the indirect part, and the conversion here, and product formulations and so on. This will be the main focus of the next years. Now I would like to hand over to my colleague who will give some details or some results on the indirect biorefinery, which is part or results from this project, but that also build further on earlier projects ongoing at VITO.
So about the fractionation of the black soldier fly, so actually the goal there is that we know that all insect fractions, so the proteins, the fats, the chitin, they are very valuable, but they are not always present in the ratios that are needed, for example, in feed. So sometimes there can be too much fat in the insect, or digestibility of chitin can be a problem. So that's why we fractionate insects in proteins, fats, and chitin. So we started here from the black soldier fly larvae. First step is a mechanical pretreatment. We obtained 2 fractions. Fraction 1, which is about 63% in mass, and fraction 2, which is about 34% in mass. First fraction contains almost all fats, and about, it's a little bit more than half of the proteins. The second fraction contains a little bit of fat still, about half of the proteins, and all the chitin is in there. So all the chitin is in 1 fraction. Also rendering, this is actually a technology well known for several years. It's being used to separate fats from animal residues, for example for fish oil, using high temperatures. Disadvantage is that you can have an effect on protein functionality. And you get a protein hydrolysate fraction, with a nutritional value, so it still contains amino acids, but the functionality of the proteins is not always intact. And we did some experiments with rendering in the lab, so we boiled the insects, and in the graph you see the solubility curve of the protein pellet, and then showing for a whole pH range from pH 2 to pH 10, you see actually a very low solubility, so about 10%, indicating that the proteins are denatured, so they lost some of their functionality. We also obtained a pure lipid fraction in this rendering, so this is the advantage. So we obtained 96% of pure lipid in one fraction, but also a lot of fat was still present in the protein fraction, which is of course a disadvantage. Also, organic solvents are often used in industry to defat, for example, oil seeds. Hexane, for example, is actually a lot been used in industry. And here we also used several organic solvents like hexane. And the result is that hexane is most efficient for fat recovery from dried insects compared to other organic solvents like petroleum ether or ethanol or acetone. But then we also looked to the effect of the organic solvent extraction of the fats and then on the protein solubility. And as you can see here, so the blue dots, these are the fraction 1. So you see that they have a large solubility at low pH, pH 2, pH 3. We have a dip at around pH 4, pH 5. And then also again a large solubility at the higher pH from 8. But for example, for the red dots, so this is with ethanol, we see that ethanol has a negative impact actually on the protein solubility. And hexane actually has less impact, and also these results are confirmed by SDS page. And then also at VITO we are developing an alternative procedure, which I cannot tell you much about it at the moment because a patent has been submitted. But briefly, so larvae are separated into fractions, and then the first fraction is further separated, further fractionated in 4 subfractions A, B, C, and D. And if you look to the protein distribution, so actually in all the 4 fractions we have a lot of proteins present. The molecular weight of the proteins is changing between the different fractions, also the solubility. And then for the fat distribution, also we have some fats with the different protein fractions. So the conclusion there is that the fats and the proteins, they are not completely separated over these 4 subfractions A to D. And then something completely else on chitin and chitosan. The last 2 days we heard a lot about proteins, also about the fats from the insects. But there is actually also a third component, the chitin, which is also very important. And so sources, as you know, crustacea are well known for their chitin, also fungi and insects, of course. So for the moment, the industrial processing of the chitin is being done by using acids to get rid of the calcium carbonate, using alkaline extraction to get rid of the proteins, and sometimes decolorization. For example, hydrogen peroxide is used to take away the pigments. Now here in the picture, you see a structure of chitin. So chitin is a natural polymer, a biopolymer of N-acetylglucosamine. And per definition, chitin is that structure where you have a degree of acetylation larger than 50%, compared to chitosan, where the degree of acetylation is lower than 50%. And then also with the blue circle and the green circle, you see the functional groups that are present in chitosan, and they can be used for chemistry and biochemistry. And this is the reason why chitosan can be an important molecule for chemical, also biochemical reactions. So when we start from chitin, first you have deacetylation to chitosan. This is often been done chemically, so using sodium hydroxide at a large concentration, 30, 40 to 60%. This first step, and these were results from the chitensec project we did in Belgium. So the first step has to be chemically because this is often explained by the solubility. The solubility of chitin is very difficult. It's a very difficult molecule. Actually, it's insoluble compared to chitosan, which is more soluble in acidic media. Also molecular weight and degree of acetylation of chitin, chitosan are important parameters to compare different types of chitosan. So then the second step, so going from chitosan to chitosan oligomers, so it's a depolymerization step, and this can be done chemically, but also using enzymes, for example. Some properties, so chitin and chitosan are biopolymers, so they can be used as a building block with a link to biodegradability. They have important antimicrobial properties. This is actually the main reason why a lot of people are looking into this chitosan. So I mentioned there a review about from the colleagues of the University of Ghent that was recently published, and they showed that the type of microorganism is important related to the antimicrobial properties. So the fungi, gram-positive, gram-negative bacteria, this can all have an effect. Also, the activity, the antimicrobial activity is increasing when the degree of deacetylation is increasing, and this was explained by the electrostatic interaction between the protonated amine group and the negative cell surface. Also, molecular weight is important, so activity increases with low molecular weight, and they defined low molecular weight chitosan between 16 and 190 kilodaltons, but also the smaller molecules, the smaller chitosan oligomers can have important antimicrobial properties. Type of chemistry can be important, and also environmental effects. So activity, antimicrobial activity is increasing when the pH is decreasing, when temperature is increasing, but there is also a link with the chitosan solubility. A third property of chitosan is also anticholesterolic properties. So chitosan can bind lipids, antioxidant properties, and also dietary fiber, so it's not digestible by a human. Some info of the chitosan markets and some applications. So in 2015, chitin production, especially from the crustacea, was about 28,000 tons, but the demand was more than double. And also the market growth in 2016 was $2 billion, and the prospect for 2021 is more than double, so $4 billion. Chitosan for the moment is a lot been used for wastewater treatment, so used as a flocculant, binding with metals, proteins. Also for biopharmaceuticals, like for example the wound healing, the bandages with chitosan in there due to the antimicrobial properties. Also pills which can have an anti-cholesterolic or a weight loss effect. In agriculture, chitosan can stimulate plant growth, can be used as a biopesticide. It can also be used to coat seeds, to help seeds during their first week when they are being spread on the field. In food, as a dietary fiber. In feed, due to this antimicrobial properties, it can be an important replacement of antibiotics, and also for some materials. And at VITO, we are working especially on the deacetylation of the chitin, but also on the further depolymerization in the different oligomers, and then the separation with membranes, for example. And this is the last slide. So this is the InDIRECT consortium. So Leen explained all the partners already. So I would like to end also with thanking the BBI for funding this project. And if you have any questions for me or Leen, we would welcome to answer them. Thank you.
I have a question about the chitosan. What is your benchmark for the chitosan extract from insect versus chitosan extract from crustacea, especially shrimps?
Well, the benchmark, it depends on the properties also, but the benchmark is a few euro per kilogram for the chitosan. Yeah.
Yes, but your cost of production are the same than chitosan extract from crustacea with insect?
We expect it for the chitosan, we expect it to be the same, yeah, in rough numbers. Yeah.
Okay, and the chitosan you were talking about is extract from which species of insect?
So in the project we are working with two insect species, so the black soldier fly and the lesser mealworm.
You are also extracting chitosan from black soldier fly?
Yes. Mm-hmm. Well, the chitin first, and then the deacetylation of the chitosan.
I think that when you harvest your larvae a little bit earlier, you won't have chitosan.
Well, the larvae that we are using, they contain chitin. So, yeah.
Thank you. With respect to the perhaps antimicrobial properties of chitosan that you showed, or expect, do you know if it's peptides which are adhering to chitin, or do you think it's really the property of this degraded part of the chitin?
Yeah, when we are using chitosan, so first we start from chitin. It already had a large purification. You first have done the deacetylation using sodium hydroxide, and so you also get rid of the proteins. And then the further depolymerization by enzymes or also by acids. So the chitosan properties that they are mentioned there, they are really based on the structure of the chitosan. But of course, you're completely right that when you look to the whole insect, proteins are really correlated also with the chitin. And as you know, also proteins and peptides can also have antimicrobial properties, as well as the fats. The fats in the insect, like lauric acid or something, they can also have important antimicrobial properties. So sometimes people are saying, my insect or my insect's meal shows antimicrobial properties. Is it then due to the protein, the fat, or the chitin, or maybe the interaction? We don't know.